Introduce you to Katalin Gothard. Dr. Gothard is coming to us from University of Arizona, where she is a professor in both the departments of physiology and neuroscience. And she's one of those very few academics who has both an MD with a postgraduate training in neurosurgery and a PhD in neuroscience. She's received an impressive number of awards throughout her career. Most notable, I think, are the awards dedicated to educating the next generation, where she received recognition for outstanding dedication to students and also for her outstanding teaching. Her main field of research is involved in unlocking the neural basis of social behavior, where she's done a ton of fascinating work in the area, where, for example, her lab is examining things such as how affectionate touch and social status is processed in the brain. And I recommend you look at her website. It's really quite for lack of a better word, touching. When you actually see, the stimuli, how they're actually are, pegging the hands of the primaries, it's really quite something. And she essentially tackles extremely big social questions with tightly controlled and elegant laboratory experiments. So we're really excited, and we're sorry you couldn't be here in person today, but we're really excited you're able to join in remotely. So please join me in welcoming Dr. Gothard, who will be speaking on a context-dependent switch from sensing to feeling in the primate amygdala.
Can you hear me?
Yeah.
Okay. Thank you very much for this invitation. I am really sorry. Oh, I hear a lot of echo. I can not speak freely because I hear a lot of echo. I'm not even sure, but I'm still there.
We are still adjusting things. Can you hear me?
Yes, I can hear you.
Yeah, it seems to be fixed now.
Okay, great. So I'll start again by saying how grateful I am for this invitation and how sorry I am that I could not be there in person. I hope that I will do some small compensation or justice to your expectations. I will tell today a story that I hope it will be important for the trainees and the students in the audience, because it's a story of turning a failure into a discovery. And I think all students need to see this. All of us in science, sooner or later, will hit a point where we have a so-called professional midlife crisis, when we look back at 10 or 15 years of work and we realize that we were missing the boat. And this is one of those stories. And also, in terms of introduction, I would like to say that I work in emotion. And this is a very strange career choice, because you work on an entity that has a very poor definition. But as a physician, or at least a former physician, I can tell you that emotion is the most extreme form of embodied cognition. And what I will tell you here, the story that I will tell you, brings back in the foreground the importance of internal states of the mind and of the brain that are not necessarily stimulated and induced by external factors, but are generated by internal dynamics. And these internal dynamics, these loops that close the processing between the internal states of the body and the mind, are, in my mind, the next frontier. I will start by telling you that touch is very important for me. Oops, I don't know whether I can advance my slides. Okay, so this is our next little snack here. I don't know how to advance my slides. Okay, I know now. Okay, so you see this picture. This picture is an orphan in Romania, in one of those horrible institutions that became part of the public knowledge after the communist regime fell. I didn't take this picture, but I was there. I was there as a student who was doing the clerkship in pediatrics, and I've seen these orphans. And it's very tightly related to why I am interested in social touch, because these orphans did not die, as we thought at that time, of antibiotic-resistant ear infections or nutritional problems. We know today that they died and they suffered because they did not benefit from the protective embrace of a caretaker. They were not touched, they were not hugged. And it seemed that later on in life, when these kids were adopted by loving and caring families and were subjected to a lot of catch-up, additional training and treatment and all kinds of therapies, they still remain with a major deficit. This deficit, it's called in psychiatry attachment disorder. And it amounted in my mind to this equation. If you do not get in a critical period of your emotional, mental, brain development, the message through the skin that the world is a safe place, that missed opportunity translates into the inability of the brain to wire up normally the social circuits of the brain, so that this person, when they grow up, they will be able to establish trusting bonds with others. So that's the motivation for the research that I will present to you. So when I started to look at touch, it was obvious that there are two kinds of touch. Here on the left is a picture of somebody shopping and touching these wonderful sweaters, and does what we call the discriminative touch, the elements of touch that translate the physical feature of objects that our fingers come in contact with a complex percept in the somatosensory cortex and further up. But there's also the social and affective touch, where through our fingers and the movements of our hands, we impart emotional social messages to each other. In primates, grooming is the most positive bond-forming behavior that there is. If I would not be worried about anthropomorphizing, I would even say this is the love language of non-human primates and of macaques that otherwise are pretty mean to each other. This is the one behavior where you see the recipient go into this positive emotional state. A lot is known about non-social, discriminative touch. Very little is known about social and affective touch, minus the background that is my personal life history. So when I started this project, I started with the question of what is the difference between the representation of touch in the primary somatosensory cortex and the amygdala. The primary somatosensory cortex, a highly explored area of the brain, both in humans and monkeys and rats and all animals that come into the laboratory, represents the physical features of the touch. We know the receptors in the skin, the forces involved, the pathways involved. But from here in the somatosensory cortex, somehow this signal have to end up in the amygdala here on the right, because the amygdala is the quintessential center in the brain where the positive, neutral, or negative valence or significance of a stimulus is extracted. Every sensory system feeds into the amygdala, and the output of the amygdala, the output of the computations that take place therein, are actually sending to the rest of the brain the outcome of this emotional social evaluation: positive, negative, neutral, socially significant, socially meaningless. So what we try to do is to monitor simultaneously with linear arrays of electrodes in the monkey brain. This is a coronal section through the monkey brain. So we lower the electrodes in the primary somatosensory cortex and in the amygdala, and expected to see in the encoding of neurons that we record from there this difference between extracting the physical features and extracting the emotional features. So I'm going to describe to you now the design of the experiment, which is going to be very important. So first, to look at neutral touch, we surrounded the monkey's face with these lock-line flexible air hoses that had a nozzle, and through this nozzle, we ejected a small airflow, cool air, not a puff, not something that would make the animal startle or blink. It was a gentle airflow, the kind that you would feel when you're outside in a nice breeze. And so the different color dots here represent the different locations, the approximate receptive fields, the size of the receptive fields of the cutaneous receptors in the face that would detect this airflow. Notice that we avoided the eyes, we avoided the nostrils and the lips. And then here underneath, I show with these colorful lines the random sequence in which these 10 air hoses plus a control that was away from the face ejected these little puffs, not really puffs, these little airflows. And on each repetition of these 11 sequence, the order was randomized. So basically what we expected to get from here is that through a repeated activation of the left upper muzzle here in gray, we would get 10 different samplings of neural activity in both in the somatosensory cortex and the amygdala. Excuse me for a second. Then, after a block of airflow, we continued with a block of grooming touch. So imagine the monkey sitting in a booth, a human that the monkey is trusting and is bonded to. This is the trusted human, comes into the booth, sits down in front of the monkey, puts on a pair of gloves that was instrumented with force transducers to measure the contact forces generated during grooming, and sits down in front of the monkey and applies, as shown here, 10 gentle grooming sweeps on the left upper muzzle, exactly where this air hose applied the airflow stimuli. And then after these 10 trials, you move to another area, the area here represented by the blue dot, and you deliver 10 grooming sweeps to the eyebrow. This is a relatively naturalistic way in which monkeys grooms. They tend to sweep the same area of the skin. And after this block of green muzzle and brown eyebrow sweeps, then there was a pause, and eventually the experiment looked like that. Airflow block, pause, grooming block, pause, airflow blocks, and so on and so. And here's your time unit to give you a sense of how long this experiment was and what the monkey experienced. It's really important that you understand the design of this experiment, because understanding the outcome of this experiment depends on understanding how these blocks were run. So if there are any questions about experimental design, please ask now. I don't hear anything.
Actually,
I hope,
we
yes?
do have a question. How about this might be awkward, but if people have questions, the only microphone that seemed to really do what we need is this one right here on my laptop.
Would you translate? Would you pass on to me the question,
So
please?
we're gonna have people come down to the laptop and talk.
Okay,
Okay.
just a clarification with the airflow block. Is it in the same location repeatedly, just like with the touch, the social touch?
No, there's a randomization. So on each sequence of 11 airflow stimuli, the order is a little different. So yes, there is that difference. Thank you. That's important.
Thank you.
Any other questions? Okay, then I will move on. So then here is an example of the type of neurons that we recorded. These are two neurons recorded simultaneously from the somatosensory cortex and the amygdala in response to the airflows. So let's take here, can you see my mouse?
Yes.
Okay, so let's take here. So this is a raster where each row of these dots is a trial, and time 0 is when the airflow went on. 1,000 milliseconds, 1 second later is when the airflow went off. And each little dot here is an action potential. So what you see here is the baseline firing rate of the neuron, summed up here below, which this we call a peri-stimulus time histogram. We sum it up here. So it's approximately 3 to 5 hertz. And then when the airflow starts, because it has such a sudden onset, we have a strong synchronization of the cutaneous receptors that become activated. And then this initial phasic increase in activity settles down to a steady state activity that lasts until the airflow lasts. And then it's followed by a very minor post-excitatory inhibition, and the neuron returns to its baseline. So this is what happens in the primary somatosensory cortex. This is exactly what we expected. In response to grooming, everything is at the same level, indicating that we indeed managed to match the contact forces and the pressures, but there's no strong onset, no phasic onset, because the humans did not poke the monkey's skin. There was a gentle laying of the layering of the pressure. So that's what is reflected by this increased slope. Now, if we move to the amygdala, the airflow in the amygdala did pretty much the same as it did in the somatosensory cortex. This is really important because it just shows that the amygdala downstream from the somatosensory cortex retains some of the properties of the firing rates that were elicited by the stimulus. So here is the phasic onset. There is a decrease and a tonic response followed by a return to the baseline firing rate. But then, excuse me, please. But then what we see is that the grooming did not elicit any response. And we did this the next day, and we found the same thing. And we did it the next day, and we found the same thing. And after 100 neurons, we decided that there must be something wrong with that monkey. So we took a second monkey, and we recorded another 100 neurons, and we got the same. And then we looked at the third monkey, and the neurons in the amygdala, like never before in my career. I have been recording in the monkey amygdala for 2 decades. I have not seen anything like this. I was convinced that we're making something really, really a bad mistake. So my staff scientist at that time, Ann Martin, summarized our data like this. So these are rasters, color-coded in terms of z-scores of firing rate. This is the onset of the stimulus to the airflow in 330 some neurons that responded either with an excitation here or an inhibition in blue to the airflow stimulus. All these neurons stopped responding when the animal was groomed. We are showing here only responses that were targeting exactly the same area of the skin. So we know that that area of the skin responds to touch because it responded to the airflow, but the same pressure, different stimulus in a different block, the neurons didn't respond. Only 22 neurons out of the entire set responded both to the airflow and to the touch. This was a huge problem, a huge headache. We scratched our heads, we didn't understand it. And then we went down to doing a couple of important controls. So first we modulated the pressure of the grooming. We increased and decreased the pressure. It had no effect. Then we blindfolded the monkey, convinced that somehow the looming hand of the groomer had a an effect on the processing of the tactile stimulation, and that's what caused this lack of response. Then we decided that somehow this had to do with the reward. And as the person who asked the question suggested, maybe the order in which these stimuli were delivered, because the delivery of the stimulus was rewarded, maybe the reward interfered somehow, and the order in which things were done interfered somehow with the response. We changed everything, there was no effect. And then we decided that there was a habituation, some kind of a passive failure to respond, a passive mechanism that would account for the failure to respond. We did all the different studies to identify a possible mechanism of habituation, and we found nothing. It was really remarkable that when we looked at the probability, the theoretical probability of these neurons that did not respond to grooming, and we compared that with the probability of these neurons to respond to any stimulus anytime, it looked to us that there was an active inhibition. This could have not been achieved in any other way but through an active process. And then we asked the question of what is happening with the animals when they are grooming naturally. Please take a minute to watch this video while I'm going to mute myself for a second. In fact, I cannot. Just give me a second. Okay, so if you watch these videos, you will see what happens during grooming. This groomee here, this monkey in the foreground, doesn't have a worry in the world. She is relaxed, her eyes are closed, her muscles are relaxed, her face is relaxed. She's completely disconnected in terms of attention and vigilance from the external world. Her sensorium is turned inwardly rather than outwardly because she relinquished vigilance and attention to the grooming partner. You can see something similar going on in the background. When we looked at the primatological literature from decades ago, it was clear that during these states, the physiology of the animal is different. The animal is in a vagal tone, which means the dominance of the parasympathetic nervous system rather than the sympathetic nervous system. For those of you who may not know, the autonomic nervous system that commands the functions of all the organs in the body has two branches. The sympathetic branch is active during states of emergency, of alertness, when energy needs to be deployed in order to deal with a situation, whereas the parasympathetic nervous system is active during situations of relaxation, of calm, when the animal is, when the organism is involved in growth and repair and not in defense mechanisms. So then we wanted to know, is it possible that we do not achieve these internal states that the primatologists were claiming that the monkeys experience when they're groomed, and this is why we failed to see these results, to see the neurons responding. So then what we did, I'd like to advance my slides, and I think that what I'm going to do, I'm just going to skip and go here. And then probably it would be easier. So then we recorded the heart rate of the monkeys. And this is one example of data set where you have the heart rate, the mean heart rate plus standard error of the mean during the airflow, grooming, and airflow block. So it's clear that the heart rate was lower during the pause between the blocks and during the grooming block. Here, there's an exception, and I can tell you exactly what happened. The graduate students knocked the flashlight off the shelf behind her, and the monkey startled. But overall, it's clear that indeed the heart rate was higher during the airflow block and lower during the grooming block, as we expect from the natural situation. Below is a power a frequency plot where we have a power spectrum of the interbeat interval. And you see this band of high power in the 0.3 hertz frequency range. And this is mostly present during the grooming, or at least more prominent during the grooming blocks. So what is this? 0.3 hertz is the frequency of breathing in monkeys. And we have a phenomenon called respiratory sinus arrhythmia. When the vagus nerve is dominating the physiological state of the body, then the heart rate accelerates during inhale and decelerates during exhale. So there is this variability in the interbeat interval, and this 0.3 hertz power band here in the power spectrum of the interbeat interval reflects this vagal tone. This is the gold standard for vagal tone in animal and human research. So what we showed then is that grooming reduced heart rate. That means it reduced the sympathetic tone. Proof for that is that if you take the 3 monkeys that we recorded from, and we look at the heart rate in the airflow, and we look at the heart rate at the grooming block, typically the heart rate goes down. There are some exceptions. Biology is noisy. Then we look at the second monkey. Here, situation is very clear, although this monkey tended to have higher heart rates, just baseline heart rates. Then we look at our monkey Cotton. He was a very high-strung, nervous animal. You can see what a large range of heart rates we see here. And in some cases, the heart rate actually went up when we groomed him. And this is the first few sessions. It took him a while to actually trust the human and learn that nothing bad is going to happen to him. And then from there on, we saw this increase in heart rate variability. No, we saw this decrease in heart rate. And then the next plot is the heart rate variability, where we show that we have an increased respiratory sinus arrhythmia, which indicates parasympathetic tone. So now, we know that our neurons do not respond to grooming, and we also know that we are grooming the animals the right way to put them in a physiological state that compares to the physiological state during natural grooming, not this artificial situation when a human grooms them. So what is wrong? Why don't we see the activity in the amygdala respond to grooming? So this is the cell that I have shown you before, but I just elongated it a little bit, because when we looked at it after 3 years and we stared at it, we found out that we were looking at the wrong thing. The amygdala did respond, but it did not respond when and the way we expected it. And I would like to know whether any of you in the audience see that there is a response to grooming in the amygdala.
No one seems to be moving.
I'm sorry, I did not understand what you said. I just heard 10 hertz. And if that's what you mean, that the firing rate here is 10 hertz compared to here that is lower, then you're absolutely right. Because instead of a phase, yes? I'm starting to hear myself again. But what we saw here, that instead of a phasic response as to the airflow, the baseline firing rate of this neuron is elevated constantly, not dependent on the on and off of the stimulus. And this makes perfect sense if you think about what emotion is. Emotion is not something that flickers with every stimulus that happens in the outside world. Emotion is a slow flowing state of the brain, and in this case of the body too, which is not so easily disturbed by the by the little flickerings of the external stimulus. In this case, maybe for what matters for the monkey and what matters for the amygdala is not an individual single sweep, the on and off of the sweeps, but the entire block where the animal knows that he's going to receive this pleasant, bonding, caring gesture of social grooming. So then we concatenated the little segments of baseline firing rate from the airflow blocks and from the grooming blocks. It became very noisy because of the boundary issues. But we found neurons like this, where the baseline firing rate, the little segments of baseline firing rate put end to end, was 1 to 2 hertz during the airflow, but was around 5 to 6 hertz during the grooming block. And then it returned again to lower firing rate during the airflow and a higher firing rate during the grooming block. And we saw neurons that did the opposite, where the higher baseline firing rate was during the airflow and the lower baseline firing rate during the grooming block. Now, let's think about this a little bit. We're talking about baseline firing rate. These segments of baseline firing rate that we analyzed is when nothing happens. There's no stimulus, there's no reward, there's no sound, there's no change in anything in the external environment. This is basically what we used to think that is immutable. This is the reference to which we align responses elicited by external stimuli. It's also important to note that because in the amygdala there are a lot of GABAergic neurons that are linked in series, the idea that a neuron increases or decreases its firing rate relative to an external stimulus or an internal state, it doesn't matter. Because if you have inhibitory interneurons in series, the inhibition of the inhibition is excitation. So in the amygdala, we see this polarity where neurons respond with increased or decreased firing rate. So once we have identified new ways in which neurons can transmit important information, but at a different time scale, we could look at the entire population this way. These are the concatenated end-to-end segments of baseline firing rates during airflow, during grooming, during airflow, during grooming for the same neurons. So we find a large proportion of neurons that show this increased baseline firing rate during grooming relative to There was an echo, but it looks like it stopped. And then there were also neurons that showed a significant decrease in firing rate during grooming relative to airflow. And there were a couple of neurons, approximately half of them or more than half, that did not show this effect. So if you stop to think about this, these are incredibly small changes because we're talking about the baseline. The baseline has to be kept within a very narrow range. And we show here that at least 45% of the neurons in the amygdala, according to these statistics, show this effect. If we make it even more stringent and we ask for an effect size to be above a certain level of 0.25 of D prime, we still retain 25% of the neurons in the amygdala who significantly change their baseline firing rate between these blocks. So this was a big and important finding. And then we verified this by doing a principal component analysis. And we took each time bin and we show that the time bins organized in this lower dimensional space are perfectly linearly separable. Moreover, we took all our neurons that were recorded and we subsampled randomly. This included the 50% or more of neurons that do not show this effect according to the neuron by neuron computation. and we subsample them and ask that population whether it contains sufficient information to decode this social context, this block from which each being came from, it was sufficient to take 134 random neurons to cross the 95% confidence interval for decoding. Each amygdala in the monkey contains 6 million neurons. There are 12 million neurons in the brain that talk on both sides, that talk to the entire brain. 134 neurons out of 12 million is a very small subsample. So even though these effects are small, it takes only 134 neurons for us to know whether the monkey was in a grooming block or an airflow block, without taking anything from the stimulus periods themselves. So this was an important effect, and we wondered what causes it? What is encoded in this baseline change? So we took the neurons. Here are the neurons that showed an increase in baseline firing rate between airflow and grooming. And here are the neurons that responded higher during airflow and grooming. And this is in the baseline. And then we asked the question, is it some kind of a reverberation of the touch that is delivered to the skin that persists beyond the stimulus itself? Or is it the mere social presence of the groomer? Could it be that this context, that the presence of the groomer gives context to the interpretation of these social stimuli? So what we did, we brought the groomer in during the airflow stimuli. The groomer just sat there, didn't touch the monkey. And what we got is a deflection of the baseline firing rate in the same direction in which the baseline firing rate would go between the airflow and the grooming block. So yes, the presence of the groomer is a critical factor that influences and significant changes in baseline firing rate. Okay, can I go on?
Yeah, keep going. It happens every now and then. I'm not sure why, but it seems to stop pretty quickly, so.
Okay. So here is a partial kind of summary of what we found. What we found is that sensing and feeling occur at different time scales. Here is the heart rate of the monkey. It goes up during airflow, it goes down during grooming. And here's the baseline firing rate. It's lower, let's say in a set of neurons, it's lower during airflow and it's higher during grooming. So what we see here is an interplay between the physiological state of the animal and the baseline firing rate. It could be that this context, the presence of the groomer, as shown in the previous slides, induces a physiological state in the monkey. And that physiological state is contributing to the change in baseline firing rate. The alternative, of course, is that the animal processes with the rest of his social brain the positive effect of the presence of the groomer. So these were two very different hypotheses. Let me state them again. Scenario one is that the social brain of the monkey says, here's my groomer is going to touch me, I'm going to close my eyes, I'm not going to enjoy this. Let the baseline firing rate change. The alternative is the groomer induces a physiological state in the monkey. And that physiological state, the changes in heart rate, of breathing, or whatever comes from the body, is ascending to the brain through interoceptive pathways. These are pathways that coordinate brain states with body states. and these interoceptive pathways induce a change in baseline firing rate. And we don't know the answer to this, but in the next 5 to 10 minutes, I will show you a new experiment that was generated in light of these results that is getting us closer. The difference between these two scenarios is enormously important. It's important for all of us healthy people because interoception and our bodily function may run our lives without us knowing it. Or the possibility is that this is false. So let me tell you now about a follow-up experiment. But before that, I forgot to sort of give you my incomplete conclusions and the new questions. So the incomplete conclusions are that grooming switches the amygdala from monitoring the external world through exteroception to signaling internal states through interoception. The exteroceptive function of the amygdala manifests in phasic stimulus-induced changes in neural activity, as we expected, while the interoceptive function may manifest in sustained changes in baseline firing rate. So the first two are solid. The third has a may, because here we need more data. So now I'm going to discuss the next experiment that was designed in collaboration with some of my students, which is a decision-making experiment in an approach avoidance conflict task. So here's our monkey, and he wears on his arm a thermode. A thermode is a Peltier machine, a piece of metal that when it sees a certain type of electric input, it will heat up very quickly in less than a second to desired temperature. And when that current is interrupted, this piece of metal will cool down to the initial temperature. So let's say it goes from 37 degrees, which is body temperature, to 46 or 48 degrees. 46 or 48 degrees Celsius is hot, but not burning hot. You can keep your fingers and your hand under a faucet that delivers 46, 48 degrees water. Your hand is gonna feel hot. It's not painful, but it's mildly unpleasant. The monkey has a button with which the monkey can turn off the stimulus anytime. We typically program the thermode for a 20-second delivery of the heat. At the same time, the moment the heat goes on, drops of juice are delivered one at a time every second in the monkey's mouth. And this is his favorite juice. Now, the monkeys are not water or food deprived. They can refuse the juice and turn off the heat at any moment. So this approach avoidance conflict task gives you a measure of how much juice is it worth for the monkey to endure the hot stimulus, or how much juice the monkey is willing to give up to get rid of this stimulus. The latency of turning off the thermode becomes a measure of this judgment, of this decision. So we did this under saline injections under the skin in the back, which they didn't even feel, because as you will see later on, we did a pharmacological manipulation. But under normal conditions, we measure the heart rate of the monkey. Here are the 20 seconds for the thermode. Here is the time of the thermode to go up, less than a second. It stays on. The monkey gets 1, 2, 3, 4, 5, 6, 7, 8, 9 drops of juice, and then he said, I had enough, and it turns off the juice, and by the 10 seconds, turns off the juice and the heat, and by the 10 seconds, everything is back to normal. So this was the choice of our 3 monkeys that we tested on this task. This is what the monkeys did at 46 degrees Celsius. Then we repeated this experiment and gave the monkey a parasympathetic blockade or blockage. So what does this mean? This is a drug called glycopyrrolate. It's a very old drug used in anesthesia. And this drug is a huge quaternary amine that doesn't cross the blood-brain barrier. but it interacts with all the parasympathetic receptors, the muscarinic receptors in the periphery. So if the parasympathetic tone, the parasympathetic input is removed from the heart will beat faster. So we know that when we inject glycopyrrolate under the skin of the back of the monkey, we have this increased heart rate. And now the question is, can this change the decision of the monkey, given that the glycopyrrolate has no effect on the thermal receptors on the skin, and it has no effect on the brain? It cannot cross the blood-brain barrier. And the answer, of course, is yes. So the monkeys who received the parasympathetic blockade terminate the heat earlier. They terminate the heat 2 to 3, sometimes even 4 seconds earlier. So what this means is that when the state of the body is changed by this drug, the animal decision-making circuit in the brain produces a different output. The only way in which this decision-making can be influenced by this drug is through interoception, probably through the vagus, but possibly also through other interoceptive pathways that go through the spinal cord. Interestingly, these pathways that go through the spinal cord are primate unique. There are pathways of interoception that are not present in lower mammals, only in primates. So we have this result. We can conclude here, based on the change of latency of turning off the heat between the saline, here's in blue, in 3 monkeys, the time when the mean, and these are all the points, these violin plots contains all the data points, that they turn it off, and the turn off during the glycopyrrolate. So clearly we show that changes in the body physiology bias decision-making. And then to enrich this data set a little bit, we had the opportunity in 2 of our monkeys to test 2 other drugs. The one in yellow here is isoproterenol, which is basically a form of adrenaline that doesn't cross the blood-brain barrier. So instead of removing the parasympathetic drive from the organs of the body, we actually increase the sympathetic drive on the organs of the body with exactly the same result. And in green is atenolol, which is a form of propranolol that doesn't cross the blood-brain barrier. Propranolol is a beta adrenergic receptor blocker used very widely in patients with cardiac problems, with high blood pressure, and also in patients with anxiety. And as you see that the mean here in the green was somewhere in between glycopyrrolate and isoproterenol and saline, because this is not changing the tone, the intrinsic activity in the sympathetic or parasympathetic nervous system. It just blocks the inputs that otherwise would activate the sympathetic nervous system. So what we have achieved here is probably, I don't like to say this, but I don't really think that anybody tried this in primates before, to manipulate the body and extract from the brain a sophisticated decision-making decision-making that involves weighing the cost and benefit of one's action. And the reason why this is really important, because emotion has these, has both the internal state, it's kind of an overlap between the internal state of the brain, the external stimuli, and how this internal state influences the processing of external stimuli. In this case, how the internal state of the animal evaluates how hot and unpleasant the heat stimulus is. And also it has to do with an action propensity. Emotions always come with an action propensity, either a defensive action or an aggressive action or an affiliative action. There is a bias towards an action. And here we also have the action of interrupting a stimulus that the animal deems unpleasant. So now we can draw some conclusions. So we can say comfortably that grooming switches the amygdala from monitoring the external world to signaling internal states. We showed that these internal states are informed by interoception, and we showed that interoception biases decision-making, biases decision-making about pleasant and unpleasant stimuli that are evaluated in the amygdala. But we didn't do our full homework yet, because we don't know whether the baseline firing rates that we showed in the amygdala to line up with internal state really depend on interoceptive inputs. And these are ongoing experiments in our lab. And ask me in a year what we found out. These animals are implanted, the drugs are ready, the electrodes are ready. We're just waiting for the students to finish the behavioral component of the project and go to the neurophysiological component. So I think this is my last slide that shows all the people who contributed to this project, former graduate students and current graduate students, and sort of senior colleagues. Dr. Fuglovend is a somatosensory neurophysiologist, by the way, a PhD graduate from the University of Waterloo. Sliman Ben Smaya, former professor at the University of Chicago, an expert in somatosensory processing, and Kevin Lin, a computational neuroscientist who works together with a mathematician, Alexa O'Coin. And what they're doing right now is showing that you don't have to get to the level of single units to find that the baseline activity contains information about the social and emotional context. The local field potentials contain that information too. Throughout this work, I have been generously funded by the National Institute of Mental Health, and I'm very grateful for that. And with this, I'm going to stop share so that we can see each other, and I'm very happy to respond to questions.
Thank you.
So a little bit more pressure on the question askers. If you have a question, you need to come down here and ask it. I'll start out this very simple question. So the thermal things on the monkey's arms, you must shave the arm first so that they can feel it. Okay.
Yes. We have to actually keep their arm in a comfortable little vat, and they Velcro there so that the contact between it's about a square inch surface, that contact between the skin and the metal plate stays the same.
Okay. And the you had the thing about the elevated firing rate in the amygdala during the grooming. Could you compare that to at the very beginning? Like it's elevated from something. Was it noticeably
Yes.
lower at the beginning?
Excuse me. So this is a very good question, because we just realized that if we're right about this experiment, that there's no such thing as baseline. The baseline is no longer this nothing happens state. The baseline carries with it information about features of the world or of the internal state that transcend the trials. So there's no point of hunting for this absolute baseline. What we did is we compared the two. So this is a relative increase or decrease. In fact, the larger implication of this is for the imaging people, because the imaging people compare the bold signal during the baseline activity to the bold signal elicited by a particular stimulus. But if the baseline itself contains information, that t-test, that comparison, which becomes the measure of neural activation in the brain, has to be reconsidered and has to be compared across multiple states, because maybe this subtraction actually steals away the real effect.
Right.
Hello, thank you so much for
Hi.
your talk. That was so interesting.
Thank
I have two questions. My first one is, so the grooming experiment. I understand that you had a tactile measure with the glove from the experimenter to groom the monkey. But in humans, for instance, mothers are told to hold their baby without any barrier, really against themselves. And so I was wondering whether taking out the glove would provide an increase even in or a longer effect of that 10 hertz modulation. Did you test that, or
Yes,
does that make sense for the monkey? I don't know. I mean, I'm kind of taking that from, you know,
it
the
does make
human
sense
literature.
for the monkey. It does make sense for the monkey. And I have to tell you that I wish this was possible. But because the monkeys are carrying zoonotic diseases, we are required by United States Department of
Oh.
Agriculture to wear gloves. We cannot touch them without gloves. Now, in another institution, where I was younger and I didn't have the responsibility of the lab, I have broken these rules. And actually, I have been groomed by monkeys. My forearm, the hairs on my forearm has been plucked and groomed by monkeys. Now I have a sleeve of very ugly artificial fur that looks like monkey fur, and I pull it over my hand and I offer it to them for grooming. So we establish this bond. But unfortunately, your very astute observation that skin-to-skin contact, it's really part of that emotional, social bonding that takes place between the baby and the mother, and between humans who just hold hands, between the nurse
Yeah.
and an elderly person who hold their hands. This is not possible because of the regulations.
And I mean, in the natural environment, obviously, the monkey is skin to skin. So yeah, I was just really wondering. Okay,
Yeah.
and my second question is about the decision-making experiment, which is really fascinating. So the stimulation that, or the effect that you're showing is really a reduction in what you could qualify as a nociceptive stimulus. What about a pleasurable stimulus?
I'm so happy you asked this. This is the kind of question that you're always hoping for. So of course, there is the possibility that what the glycopyrrolate did, that the increased relative sympathetic tone did, was either it increased the sensitivity to the nociceptive stimulus or decreased the sensitivity to the reward. So what we did, when I have a slide, but I'm not going to bother, I'm just going to tell you, we have a task where the monkey had to make decisions between good things. It has to decide whether he wanted blueberries or blackberries, or, you know, kale or iceberg lettuce. And we verified these choices a priori before the injection was glycopyrrolate. And we wanted to see whether, one, the ordinal sequence of animals' choices changed, that he would, so for example, maybe he becomes thirsty because the salivary glands would not produce saliva and would produce something that's juicier, would prefer something that's juicier, or would change its preference or would not care about. There was absolutely no change. So this manipulation did not alter the approach choices, did not alter the valuation of anything that was positively deemed before the intervention. It only reduced tolerance to stimuli that required avoidance.
Okay, thank you so much.
Thank you. Very good questions.
You have one coming?
Hello.
Hello.
Thank you for this very interesting talk. I have actually, it's more a clarification question about the baseline period, because I was wondering a little bit like Jeff, like when did you measure the baseline? Because if you measured it before the stimulus, I was wondering, how did the monkey know if it was going to be a grooming or air puff trial? And if it's because they kind of learned the sequence, did you try to randomize the order of the experiment, and then did that affect the way you measured the baseline?
Okay, that's a very good question. And this is why I said that the experimental design is very important. And obviously, I didn't explain it very clearly, but I'll try again. So there were two kinds of baseline. The baseline between the blocks, when the monkey was sitting in the booth, either alone or or the groomer was going in and out, when no touch happened. And so those were times when something was still happening, because maybe the groomer was just about to enter, or maybe the groomer just left, but we kept those baseline too. The more important pieces of baseline were the periods between the stimuli within a block. So in a block, there were 110 airflow stimuli. Between each second of airflow stimulus, there were 3 seconds when nothing happened. So it was 1, 2, 3, 4, 1, 2, 3, 4. And the 2, 3, 4 is that those are 3 seconds when nothing happened. We took the middle second from that 3 seconds and we considered that the baseline. For the grooming, it was the same. So let's say I'm the groomer and I'm the monkey. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. And we take the middle second. That is 1 second past the end of the grooming and 1 second before the beginning of the next grooming. So this is a period of time that is neutral. Nothing is happening. So we took those, cut out those little periods of time from the neural activity, and we put them end to end and generated this longer period of time where we now can measure the baseline firing rate.
Okay, so this is really, really helpful, because now it kind of clarifies to me how you evaluated the baseline. So just like one short follow-up question, how much do you think that this has to do with the general arousal of the animal, or some general attentional state of the animal?
Yes, so it has to do with both. So as I showed you during the natural grooming, the video during the natural grooming, the arousal state of the animal goes down. And we have 2 measures for that. One is the behavioral measure. You see the animal relaxing, closing its eye, relaxing its muscle, completely abandoning all worries about the world. So that's kind of the behavioral reduction of vigilance, attention, and general arousal. And the physiological measure is also a very powerful measure of arousal, because the sympathetic activity, which is associated with vigilance and attention, goes down, and the parasympathetic activity, which is associated with disconnecting with the external world, goes up. So yes, this internal state reflects a general mind-body coordinated state of lower arousal, lower attention, lower vigilance, sort of like a meditation or just a state of bliss in which the monkey goes when it's groomed.
But the baseline goes up.
The baseline goes up. There are neurons in which the baseline goes down, but as I said, the individual neuron doesn't decide where the global baseline is, because individual neurons can mutually inhibit each other and can inhibit each other in series. So what we see in the local field potential is a change in power of multiple frequencies in the local field potential. So it's the up or down, it's less important than to trust that this is a big change in the neural dynamics that takes place within the network.
Okay, thank you so much.
You're welcome.
Question.
You've very nice talk. Thank you for that.
Thank you.
I liked very much the focus on grooming, but grooming is a very positive affective context for the animal.
Yes.
And the physical kind of just the physical detection of the air blowing is more of a neutral affective state. So I just wonder how much of it is due to the kind of grooming itself as a socially intrinsically rewarding stimulus in setting the context for what the amygdala is doing. Might you have a baseline increase generally with positive affective context compared with a neutral context, or maybe even different baseline with some kind of negative affective context?
Excellent point. Excellent point. I think your answer, you already have your answer because you figured it out yourself, that it is very difficult to imagine a situation of parasympathetic tone that is associated with a stimulus that has negative valence. It would be very difficult for the heart rate to go down steadily. Maybe the heart skips a beat, that's a different story. But to steadily stay down, and for the animal to close its eyes and relax its muscles in response to a negative stimulus, this would be very difficult to imagine. I think it's impossible, but one should never say that something is impossible. So you're absolutely right. It is a way of the amygdala to signal to the rest of the brain a positive affective state. And the amygdala is the most interconnected structure in the brain. It has massive projections to the entire cortical mantle. And what is really interesting is that these projections, when we look at projections between adjacent cortical areas, let's say V2 to V4, V2 talks to V4, which is higher level processing, and V4 sends feedback to V2. So the basolateral nuclei of the amygdala, for all intents and purposes, are cortex. Evolutionary, they come from the same area where the cortex is coming from, so they're cortex. But they violate this rule because instead of sending projections back only to the cortical areas that immediately send input to them, they go all the way back to V1 and to S1. They project glutaminergic excitatory projections and synapse on layer 1 and 2 of the cortex at the earliest stage of processing. So basically, there's a mesh of glutaminergic input coming to layer 1 and 2 in the cortex when the animal is in a particular state of the amygdala. And we can imagine how in this positive state, the amygdala generates a sustained level of inhibition of responding cortically to the stimulus, whereas in a state of sympathetic arousal and negative affect, when the animal has to be ready to mount defensive behavior, this input is not there.
Yeah. So I was wondering if you're willing to generalize that even further, so even some kind of momentary stimulus that's positive, negative, or neutral would affect the kind of baseline firing background, because your results didn't directly show that, but it's kind of a direction pointed to by what you've done.
Yeah, I don't know what would happen with a temporary positive stimulus, whether they would have the same effect. And the reason why these experiments are incredibly interesting to entertain, but are difficult to realize, is because if indeed this amygdala state is linked to the internal state of the body, we have a latency problem. The heart rate cannot jump up and down with the same frequency with which a stimulus can change its valence. You can't so quickly go to heart rate. You need at least 3-4 beats. And the autonomic nervous system is not designed for this flickering up and down. So one would have to still do some kind of a compromise of considering a prolonged state that is induced by positive and negative stimuli so that it can be reflected in these bodily functions. And in fact, you know, if I may, it's very easy to go from a neutral state or from a positive state to negative state, and immediately jerk up the heart rate, change respiration, change pupillary side or so. Now try to do that with the same speed when in the other direction. You can't do it. It takes a while for this echoing kind of persistent sympathetic arousal to wane away and for the parasympathetic state to come in place. This, you can't calm down. Even if you got startled and you realize it's not a rattlesnake, it's just a twig on the floor, and your dog is not going to get bitten, even though you immediately realize that you made a harsh kind of a bad judgment, your heart rate still is going to pump for at least a minute.
Okay, thanks. There'll be, I guess, a lot more to discuss, but I'll let other questioners come.
Thank
so
you.
much.